So now we are going to see a unique procedure which is called as serial dilution procedure for isolating samples from isolating microbial samples from any given type of sample, like soil sample or any garbage sample or whatever, food sample, anything like that. So from that, how to isolate your different microorganisms. So that is so simple procedure is you have to first perform serial dilution. After that serial dilution, with that diluted samples, you can plate it in your plates, the LB plates that you have prepared. So today we will see a simple experiment on serial dilution. Okay. So in this serial dilution, you require the basic requirements like your brain solution, right? So that is nothing but your salt, sodium chloride solution. 0.8% of sodium chloride has been taken, and then you have prepared a brain water solution, right? So this is your saline solution of 0.8%. That means for 100 ml, it is 0.8 grams you have to measure. Then your soil sample, wherever you have collected your soil, you have to take that soil in an appropriate container. So this is a sample container containing your soil, right? And then a spatula, and then test tubes with brain water, with saline solutions, right? So 9 ml, 9 ml each. So all the test tubes have 9 ml. So 8 tubes you have now. And then a pipette, micropipette. So these are all the basic requirements of performing a serial dilution procedure. Okay. Now, first thing is you have to weigh appropriately 1 gram of your soil sample. So we have already weighed it in weighing balance. So the soil sample, 1 gram is there. Now suspend the 1 gram of soil sample in the beaker that is with saline water. So we are suspending it in a 10 ml of saline solution. So you can just put it and then gently mix it so that it will get completely suspended. Okay, now it is completely suspended. So once you are mixing, that time itself you have to take 1 ml of the sample. So it is already 10 times diluted, right? So it is in 10%, it is in a 10% solution, right? So 100 ml for 10 ml you have added, sorry, it is in 10%. 10 ml for 1 gram of your soil sample. So it is already in 10%. So then you can make sure that it is completely suspended. So once it is suspended, then you can immediately, you should not allow it to settle down. So you can immediately take another 1 ml of your sample, then put it in your first 9 ml test tube. Okay. So already it is 10 times diluted. Now again you are taking 1 ml and you are putting it in another test tube of 9 ml brain. So now it is completely, you have added 1 ml of your solution sample. Now it is again 100 times diluted. Already beaker over 10 times diluted. So either 1 ml at the 10 times diluted. So 10 into 10 it is 100 times diluted. Then again from this 100 times diluted sample you are taking another 1 ml. So mix it completely. Then from this you take another 1 ml. And add it in the next test tube. This is another 9 ml of saline solution. So then mix it completely. Mix it by 1 or 2 times. You pipette it out and pipette it down so that it will be completely mixed. Then now if you see, it is again serially diluted for another 10 times. So first time it is 10 times you have diluted, another 10 times, so it is 100 times diluted. Then again you have diluted it for 10 times, then it is 1000 times diluted. Then again you can take it from that. That is 10 to the power 3 times you have diluted. Then again 10 to the power 4 times you are diluting it. Mix it. So it is 10,000 times diluted, right? So now you can take another 1 ml from this 10,000 time diluted sample and put it in the next test tube containing 9 ml of saline. So now it is 1 lakh time diluted. 10 to the power 5 times it is diluted. So again mix it completely and then take 1 ml and then you can add it in the next test tube containing 9 ml of sample. So here you have 10 to the power 6 times diluted. Okay. So then again you can take 1 ml, 10 to the power 7 times. You are diluting your sample. So, bacterial count, you are logarithmically, you are reducing the count of bacteria. Correct? So, next 1 ml you can take it. So, every time when you transfer the sample, you have to mix it completely. The samples will not be equally, it is not diluted. Equally, it is not serially diluted, it means. So then again take 1 ml. Then you can transfer it to the next one. So this is the final 10 to the power 8, I hope, right? So this is 10 to the power 1 times you have diluted, 10 times, that is, this is first one is 100 times, then this is 1000 times diluted, 10,000 times, that means 10,000 means it is 10 to the power 4 times, 10 to the power 5 times, 10 to the power 6 times, 10 to the power 7 times, 10 to the power 8 times, 10 to the power 9 times it is diluted. Okay, so with this 10 to the power 9 times diluted sample, you can plate. So now we have 5 plates, so 1 plate for control, so remaining 4 plates, that is 10 to the power 9, 10 to the power 8, 10 to the power 7 and 10 to the power 6 are the samples that we are going to culture it in your Petri plates, the LB Petri plates that we have prepared. Okay, so this is a simple serial dilution procedure. Okay, this is a very important question is, why we have to use this saline? we need not to depend on normal distilled water. Why we have to depend on saline? Because the cell should not be exposed to some osmotic damage. Because the cell there will be some salt concentration. If you put the cell immediately inside a distilled water, what will happen? The distilled water or salt concentration is 0. உங்கள் செல்லுக்குள்ளே இருக்க சால்ட் கான்சன்ட்ரேஷன் இஸ் சம்வேர் அரவுண்ட் 8.8%. அப்போது the செல்லுக்குள்ளே இருக்க வாட்டர்லாம் என்ன ஆகும்? இட் வில் ஸ்டார்ட் யுவர் சால்ட் மாலிகூல்ஸ் வில் ஸ்டார்ட் எக்ஸிட்டிங் அவுட் ஆஃப் யுவர் செல்ஸ். ஸோ அப்போ வாட் வில் ஹேப்பன்? இல்லைனா ஆர் அதர்வைஸ் இது எப்படி நடக்கும்? தி வாட்டர் மாலிகூல்ஸ் வில் கம் இன்சைட் யுவர் செல் ஸோ தட் இட் ஹேஸ் டு ஈக்குலிபிரேட் த சால்ட் கான்சன்ட்ரேஷன். Now once it do that, the cell will start bulging out and the cell will die. Appo you have to give an appropriate salt concentration for the cell to survive. So that is what you are using 0.8%. இதெல்லாம் நீங்கள் படிப்பீங்க. In further upcoming semesters you will learn about this hypotonic solution, hypertonic solution, isotonic solutions லாம் you will study. Okay, அதனால தான் to maintain the osmotic, to not give osmotic shock to the cultures, we are using this 0.8% saline. Because 0.8% தான் நம்ம செல்ஸுக்குள்ளே இருக்கக்கூடிய சால்ட் கான்சன்ட்ரேஷன். ஸோ வெளியே இருக்க சால்ட் கான்சன்ட்ரேஷனும் சேமு, செல்ஸுக்குள்ளே இருக்க சால்ட் கான்சன்ட்ரேஷனும் சேமு. ஸோ the cell will not get damaged. That is what we are using saline solution. Okay, now you have prepared your serially diluted samples. Now we will go and check our Petri plates. Let me see whether it is solidified or not. So after it gets solidified, we can start inoculating it by spread plate technique. Okay, so எப்படி spread plate using an L rod, how to spread your samples in your Petri plates, prepared Petri plates, நம்ம எப்படி பண்ணுறதுங்கிறத we will see it now. Okay.